This presentation is PBPK modeling of antibody-drug conjugates, concept and application. Our speakers are Dr. Felix Stadter and Mr. Himanshu Mishra. Felix is currently a senior research scientist at Certara UK. He studied biology and pharmaceutical science in Münster, Germany, and did his doctorate in Basel, Switzerland. At Certara, Felix has worked extensively on the biological models of the Simcyp Simulator, including antibody-drug conjugates and simulating therapeutic protein disposition in pediatric patients. Himanshu is currently a director of business development at Certara's Simcyp division. He holds a Master's of Business Administration and a master's degree in pharmacology. He previously worked at Certara as a research scientist supporting the development of the Simcyp Simulator. Felix and Himanshu, welcome to today's event. I'll now turn it over to Himanshu to begin the presentation.
Thank you, Susan. Good morning, good afternoon, good evening, everyone, and welcome to our webinar on PBPK modeling of ADCs. So today, over the next 40 odd minutes, we'll cover the basics of why we are talking about ADCs and present some relevant examples around modeling of PBPK and help how it can help you accelerate your ADC development programs. So, the first ADC was approved almost 20 years ago by FDA, and since then 12 more ADCs have been approved, and majority of them have been in the last decade or so. And as you can see from the graph on the right, the proportion of ADC approvals is increasing, from the FDA itself. And so what is an ADC? Next slide. Yes. So what is an ADC? So ADC is a combination of antibodies and cytotoxic drugs. Antibodies, which are low potency and high specificity, combined with a high potency, low specific cytotoxic drug, give you a high potency, highly specific ADCs, which essentially improve your therapeutic window. ADCs together have, the antibodies function is to work as a target recognition at the tumor site. And then we have what we call as a payload, which comprises of the linker and the small molecule cytotoxic drug. Some of the key terminology that you have to keep in mind is drug to antibody ratio, or DAR as it's called, is the average number of drugs conjugated to the antibodies. It can be anywhere from 1 till I think 7 or 8. Deconjugation is the release of small molecule payload from the ADC by chemical or enzymatic process at the site of action. Ideally, catabolism of ADC is also accompanied with payload release. Okay, so why are we talking about PBPK modeling? Because there's a lot of questions that we need to answer when we are looking at ADC development programs, and essentially what is the impact of molecule changes on the pharmacokinetics? Which pathways drive deconjugation? What are the DDI potential of the payload? And what's the concentration at the target site? Because that determines your PD or the toxicity effects. How can off-targets effects be explained? And what is the distribution in different special populations? So these questions and many more can be answered using our Simcyp PBPK simulator. One of the key aspects here is simulating both the large molecule, the antibody, and the small molecule is simultaneously, because that's what drives the PK and the PD link. So to talk through our model and relevant case studies, I'll hand it over to my colleague Felix. So Felix, over to you.
Thank you. Okay, so now we will look at specific case studies that we have done within the Simcyp simulator using our ADC platform. So what we're seeing here on this slide is our very general framework for our PBPK model, a very technical drawing of the body by different mathematical compartments represents organ and tissues of the body. Now we can parameterize those with physiological information of several different species. So here as one example, we can utilize preclinical data where we have a lot of information of our ADC, for instance, tissue concentration. We inform the model structure by the physiological parameters of the animal species, such as rat or monkey. We use the extensive preclinical data to learn more about the ADC drug, to confirm the model, to improve the prediction, for instance, of the tissue concentration. And then what we can do is we can switch the physiological database that informs the model structure to human and translate the model to humans and apply it in humans. And we can also think about this of having clinical data for healthy volunteers, where we inform the model structure by physiological data for healthy volunteers. We verify the model of our ADC, and then we switch the physiological database to another ethnicity, such as Asians. We switch it to people with hepatic impairment, renal impairment, or obese. So we can also use the simulation for simulating the drug disposition in special populations.
There you go.
No, it's working. Okay, so how is our proposed workflow for these bridging studies? Well, first of all, we usually start with IgG, because there are many unknown parameters in several of the cellular processes that cannot be measured and have not been measured, where we need to do calibration steps. And we usually use IgG for this at Simcyp. So we start simulating IgG in the animal species. And then we move one step further in simulating the antibody part in the animal species by assuming that an IgG-based antibody, with switching parameters that are different between IgG and the antibody, like binding affinity to FcRn, molecular weight, we will be able, with the calibrated parameters, to simulate our antibody in the animal. And then we use information that we have for the release of the drug about deconjugation, for instance, or if it's a non-cleavable linker, we're using then the lysosomal catabolism to estimate the release of the payload. And we try to match then the total antibody, conjugated antibody, and release drug in the animal species in plasma. And then we can go one step further in looking at tissue concentration that might have been measured in animal species, verify the animal model, calibrate the animal model if necessary. And then we take the physiological database of humans, translate the model to humans, ideally with clinical data in plasma of healthy volunteers, and then we can apply the model to other special populations, to other scenarios like for DDIs. As I said before, we need to have a lot of information to inform the model structure. And I just listed some of the information here that we need for large molecule drugs, for ADC drugs, such as lymph flow, FcRn expression, endosomal volume, and many more. By knowing the species differences, we can do those kind of bridging studies where, as I said, the model structure will always be similar, but we're changing those kind of parameters to translate to different kind of scenarios. And one example is shown on the next slide, where we had the question, can we predict any ethnic differences for an ADC? So this publication looked at trastuzumab emtansine, and it dealt with 3 different ethnic groups: people of European heritage, white, Asians, which were mostly Japanese, and a third group classified as others. And the question was really, can we replicate these findings of the study within the Simcyp simulator? Can we use PBPK modeling by exactly what I said? We're switching the physiological database and the model structure to reproduce the clinical findings, and can we then predict the ethnic differences? So what you can see below are the modeling results on the left-hand side for the conjugated antibody and on the right-hand side for the released drug. What we're looking at here is the ratio between Asians and North European Caucasians. So on the left, to the conjugated antibody, we see the peak concentration to the left, the AUC extrapolated to infinity in the middle, and the clearance on the right. And what we can see is that in white, the white data is the observed, and the gray bars in front is the predicted. And we can see that we are able to predict the differences between Japanese and North European Caucasians for the conjugated antibody, particularly looking at the AUC in the middle. We are also predicting variability. However, I have to say here in this case that the differences in things like lymph flow FcRn abundance are unknown, so we're using the same parameters, but you can see from the observed data that there's essentially no differences in the conjugated antibody, and that's what we're predicting within Simcyp. However, for the released payload drug there, it becomes important all the differences that are known between ethnicities concerning, for instance, metabolizing enzymes. And we can again see that we are able to predict the peak concentration that was given for this ADC. So thinking it further, we can utilize the PBPK model in simulating the released drug properties in different special populations, not only ethnic groups such as Asians, but also in hepatic impairment and renal impairment. To get more confidence in the antibody part, I think we need to have more verification studies and more verification steps, but this is certainly also an area that will grow and develop over time in the future. Another case study was published in 2020 where the Simcyp Simulator was used to inform the label for DDI studies. So what has been done here is that a PBPK model was developed for brentuximab vedotin. It was verified against clinical data in plasma. And a DDI study has been conducted to the small molecule drug MMAE, which is metabolized by CYP3A. And the DDI study was done with ketoconazole as a strong inhibitor and rifampicin as a strong inducer of CYP3A. And this DDI study, this clinical DDI study, has been used to verify the PBPK prediction. So there was a fully verified model for this ADC, including the DDI part. And then they had a second ADC, where there was only clinical data available for the plasma concentration of the antibody and the released drug, but there was no DDI study. And essentially, the PBPK model was developed for the ADC, was verified against those plasma concentration, and without running any clinical study, the Simcyp Simulator was used to predict the DDI with ketoconazole and rifampicin as strong inducer and strong inhibitors. And this simulation was used in the drug label, informed the drug label without doing any clinical study. So the Simcyp Simulator waived, in this case, the study. And this is really the heart of what we're doing in the DDI prediction. So to go a bit more in detail, what we're seeing here on the left-hand side are the prediction results for the conjugated antibody on the top and the released drug small molecule at the bottom. And what we see in the table, in the upper table, is the results for the first ADC, brentuximab, with ketoconazole and rifampicin. We see the prediction versus the observed data for both scenarios, the strong inhibitor and strong inducer, and also if the small molecule drug is the perpetrator, where midazolam has been used as the victim drug. And then in the table below, we can see the prediction results when we are using the second ADC having the same linker construct and the same payload, verified against clinical data to the right, and we can see the prediction for the AUC ratio, which informed the drug label. And it is also very promising that the FDA has released the new guidelines for antibody-drug conjugates at the beginning of this month, where it's explicitly mentioned that for the DDI studies, for the small molecule drug, PBPK modeling can be used. So this is a lot of potential in saving clinical study and using PBPK modeling at its heart. Okay, now after looking at the case studies, we're now going into the model structure, what we're using in the Simcyp Simulator. So what you can see here on these two slides are our distribution models in general for large molecules that we also use for antibody-drug conjugates. So on the left-hand side, we see the minimum PBPK model, where all the organs and tissues of the body are lumped into one lumped compartment. And given the size and the polarity of large molecules in general, we have several different subcompartments: the vascular space, endosomal space, interstitial space, and the intracellular space. And we train lymph flow into a central lymph node compartment, which brings the fluid to the central blood compartment and then to the vascular space. To the right-hand side, you can see the full PBPK model, where different compartments represent organs and tissues of the body. We are having the same subcompartmental structure for each of the compartments. we're draining lymph flow from each compartment, collecting it in the central lymph node compartment, and bring it, excuse me, and bring it to the venous blood pool. So there's flexibility in using one of the two distribution models, whether we are interested in reproducing or simulating a quick example using the minimum PBPK model where we want to explore certain issues, or whether we want to see understand the concentration in certain tissues. So then we have to use then the full PBPK model, but this flexibility is given within Simcyp. What is also very important is that we are using a lot of science in order to develop our model. So this is really one of the key points that we're doing at Simcyp, and also for large molecules and ADCs. And I just want to bring one example, and this example is about the distribution of large molecules. So if you think about injecting our ADC, our antibody, into IV, into the blood vessels, our target might be a membrane-bound receptor in the interstitial space, so we somehow need to have distribution between the blood vessels and the interstitial space. So the hypothesis is that we have a set of pores in the endothelial cell layer, which is a barrier for those large molecules. And that we have flux of fluid and proteins through those pores between avascular and in the interstitial space. Now, the issue is that these processes cannot be measured. We cannot just take the endothelial cell layer. We cannot just look what is the size, what is the amount of those different pores. So it is a hypothesis in this way. So what we have done is to collect data for the interstitial space concentration and the plasma concentration of proteins of various different sizes, to use those to calibrate the parameters of the two pore. That means the size of the pores and the amount of the pores. And you can see those results are to the right-hand side, to the top for the muscle and at the bottom for the liver. And you can also see that they are distinctly different when we're looking at proteins with a higher hydrodynamic radius. So there's a higher interstitial space concentration in the liver, which has to do with the blood vessel structure, that you have large gaps between the vascular and the interstitial space for the liver, which is reflected in the model. So as I said, this is just one example where we try to understand the physiological processes that drive those fluxes, where we try to understand the mechanisms and where we try to parameterize those mechanisms. And if those data aren't available, then we try to find a scientific argumentation in order to calibrate those parameters, which is shown here for the two-pore hypothesis, which we also do for the monoclonal antibody part. Now, after looking at the large molecule models, we are now introducing the ADC, and we can put it into one sentence. We can say we simulate the large and small molecules simultaneously. Sounds very simple, but it's really difficult to develop those kind of model, and needs a lot of technical details, and that needs a lot of understanding. But essentially what we're having is that our antibody-drug conjugates are a heterogeneous mixture of different DAR numbers. So that means here in this example, in the middle, we can see we're having up to a DAR number of 8. So we have 8 payloads attached to the antibody. And the different DAR species, they are linked via the release of the drug via several different pathways. So we can go down to DAR 7, DAR 6, etc., etc. And all those processes happen in parallel everywhere. The released small molecule drug is then taken into the small molecule PBPK model. Is using the full PBPK model for small molecules. And the idea is, in general, if you have, for instance, drug release in the liver via deconjugation in the interstitial space, via catabolism in the endosomal space, or because there is the target is in the liver and we see drug internalization with subsequent degradation, the released small molecule will be automatically be in the liver as a metabolite of the conjugated antibody. Will be immediately available for transport by any active transporter, will be immediately available for metabolism by any of the enzymes that we have in the liver, and will be immediately available for DDI, if there's any co-medication, if you have any kind of inhibitor or inducer. So this is the general idea of the model, that we have the simultaneous measurement, and this really allows to see the concentration of the released drug in several different tissues in real time. This is a very complex and technical drawing, but the essential point of this is what I've already said, that we have to link those two models. So we have our full PBPK model for ADCs here in the to the left, and we're releasing the payload directly into the compartments for the small molecule simulator, where I said it would be immediately available for all the different processes like transporters, DDIs, but also for any kind of pharmacodynamic model in any of the different tissues and compartments. So this is really helpful to see if the concentration reaches a significant amount to have efficacy, to have any kind of effect, but also to understand any kind of toxicity, to see if there's any release in a certain organ reaching a high level of the small molecule drug, which might explain toxicity in this organ. What I've already said is that we have release of the drug by binding to the target. So we consider target binding in the ADC model. Essentially, the target can be placed in plasma, in the lymph nodes, in the interstitial space of any compartment that we have, of any organ and tissue, and also in our tumor model, so that we can predict the target binding of the antibody of all the several different DAR species to the target by knowing then the association and dissociation rate constant, or by using any of the approximations of the TMDD model that we have. Very importantly, the internalization, as I said, will release the small molecule payload, will release it in the full PBPK model for small molecules. What we're also having is a tumor model for both the large and the small molecules. So again, if we are using the tumor compartment in the PBPK model and we have a release of the small molecule drug, it will be immediately available in the tumor compartment of the full PBPK model for small molecules, and can be available for any kind of pharmacodynamic model that might be necessary to lead to this. And now we're looking at some of the simulations of the ADC model. So what we are having here is a very general simulation, where we assume that we do not have any kind of deconjugation, we don't have any target binding, we don't have any internalization. The release of the small molecule drugs comes only by non-specific catabolism in the endosomal spaces, in the lysosome of the endosomal spaces. So what we're seeing, as we have our simulation to the left, we have our simulation of the several different DAR species. They are perfectly in parallel to each other. The part of simulating different DAR species is that in a very high and abstract level, you can look at this simulation, and you can also think about simulating two maps that are administered simultaneously. So it provides a very general framework to say, in order to use this kind of simulations, so we can also look, if we administer two maps that bind to the same target, what would be the effect? So this is what you can see here by looking at these simulations where they run in parallel. In the middle is probably something that's more frequently used, where we see our total antibody, conjugated antibody, and the free drug. And here you can also see in the simulation that the free drug runs in parallel to the conjugated antibody. So to the parent drug, if you want, the small molecule drug is then treated as the metabolite. And to the right-hand side, you can also see the prediction of the distribution of the small molecule drug in several different tissues. So this is what I said before. We can simulate and predict the tissue concentration. That's the beauty of a PBPK model, of a full PBPK model, that we can link those kind of concentrations also to efficacy or to toxic effects by adding a PD model after the PK model. In the next step, we perform the simulations to understand the deconjugation. So we're adding now deconjugation in the plasma and in the interstitial fluids, and in general in the extravascular space. So if you look to the figure to the left, we can see an accumulation of the naked antibody. As eventually all of the different DAR species will reach to this naked antibody. Again, in the middle, we can see that the payload is treated as the metabolite. It runs perfectly in parallel to the conjugated antibody. What we can do by using the simulator, that's what Himanshu mentioned at the beginning, is that we can check for what happens if we're using different linker constructs, what happens if we're using a non-cleavable versus a cleavable linker, what happens if we're using a different kind of cleavable linker, what happens if the deconjugation rate changes, if there's no deconjugation rate. So we can predict those kind of scenarios. We can predict how it affects the pharmacokinetics of the released small molecule drug, how it affects the pharmacokinetics of the ADC. So this would be one utilization of the PBPK modeling approach. And here on this slide, we have two different scenarios. So to the right, we again talk about deconjugation as well as catabolism, which is both used in here. So at the top graph, we have an immediate release of the small molecule payload. So again, we see that the small molecule drug in the green light is in parallel to the conjugated antibody. And at the bottom, what we see here is that actually the green line is no longer in parallel. And this is due to a delayed release of the small molecule drug. So if there's any kind of catabolism in the endosomal space, it might be that the small molecule drug is not immediately released after the catabolism. It's not immediately presently available in cytoplasma for any kind of action, or is not measured. So you see this kind of divergence between the parent drug, the conjugated antibody, and the small molecule payload. And you can use modeling and simulation to understand those kind of effects and to also see how these affect the pharmacokinetics and the release of the drug and the tissue concentration. What we see on the figure to the right is how the affinity to the target changes, because eventually we want to have an antibody that binds with high affinity to the target, and we can also run simulation. What happens if we change the affinity? What happens if we change the target, for instance? What happens if the internalization rate changes? How does it impact the release of the small molecule drug? How does it impact the overall pharmacokinetics? So this is just a very general overview of the Simcyp simulator and what we can do in terms of predicting, as I said, linker stability, the release of the payload, different routes of the release of the payload. What happens if we are switching differences in the target binding? So overall, I hope we could show you today that PBPK modeling can be used as a case example in order to extrapolate to different populations, like we have done it for the Japanese population. But we see here really a growing area in the future where we can use all kind of disease population, where we can use other special population like obese subjects. That might be very helpful in understanding the pharmacokinetics and understanding the disposition in those special populations. We have also seen that we can use the Simcyp simulator to predict the drug-drug interaction for the small molecule drugs, and we can use those PBPK simulations directly in the label without running any additional clinical study, really waiving the study, saving time and money. We have seen that we are simulating the small and large molecules simultaneously, so that has the advantage that the small molecule drug is immediately present where it's released. We're getting the right tissue concentration. We can understand the efficacy. We can understand toxic effects. We can link it to PD models, to any kind of pharmacodynamic models that we have. We have seen the flexibility in the model structure of using minimum and full PBPK model, using different TMDD models, using the tumor model. And we have seen, as I said before, that we can use the Simcyp Simulator to predict the disposition in special populations. And with that, I'd like to thank you for your attention.
Wonderful. Thank you so much, Felix and Himanshu. So I can see we've got some questions from the audience. So one person says, if we go a step further for ADCs that share the same linker and payload, do you think that there's even a need for PBPK prediction? The examples you showed in the case study showed almost identical outcomes. What do you think of that?
Yes. I think the requirement from the regulatory would still be to have any kind of data, any kind of clinical data or about the PBPK modeling. But I agree, if we're using the same linker and the same payload, we will probably always predict the same DDI, so we will really have this experience. And if the regulatory agencies agree with that and accept it, then yes, you can go that way. I mean, this is something where we have a lot of experience with cytokine. The effect of CYP3A4 from cytokine release, because we had now several examples, it always comes down to the same. So if we're having this kind of information, yes, but we have to think it further, because this was one example for one linker and for one payload, but there will be different linkers, there will be different payloads, and we can still utilize the PBPK modeling for all those new kind of scenarios, because you don't want to stick always to the same drug, right?
Right, makes sense. Someone wants to know, after you inject the ADCs, how fast are the ADCs diluted by the blood volume, and how homogeneous is the dilution?
In general, that would be, I guess that would be very quick, and it should be homogeneous in terms of the average DAR number. It might not be homogeneous if you're looking at individual DAR species.
Great. So your presentation talked about modeling ADCs that are linked to small molecules. Is it possible to model an ADC that's linked to a small interfering RNA?
Yes, generally, yes. We will develop a model for oligonucleotides in version 23, so that will become available in 1 year time. But currently, you could still, I think you could still probably trick the system and still model your oligonucleotide. Definitely possible, but as I said, version 23, there will be an oligonucleotide model, and then you have the full capability to simulate such a scenario.
Great. Stay tuned, folks. Exciting things coming. Can you tell us what data you need to build a PBPK model for an antibody-drug conjugate?
Yes, so this is actually the most complex scenario in terms of data, because you need to have both data for the large molecule and data for the small molecule drug. So for the antibody, it's the molecular weight or the hydrodynamic radius, where we predict all the parameters for the 2-PAR hypothesis that I showed you. We need to have an idea about the binding affinity to FcRn, an idea about the clearance pathways. So what I mean with that is, if there's a lot of unspecific binding happening that we need to consider, we probably need to have an idea about the charge of the antibody, because that can affect how the uptake is into endosomal cells. And we need to have the information about the target. So what is the target abundance? Is it expressed throughout the body? At which concentration? And we need to have an idea about the affinity, the binding affinity to the target. So that would be for the large molecule. And then for the small molecule drug, you will need to have the information again about molecular weight, lipophilicity, as the pKa fraction unbound, and also the information about the metabolism.
Great, thank you. Looks like we've got one more question. Someone says, regarding simultaneous simulation, what's the likelihood of the payload being cleaved off one small molecule at a time, or all at once, or 2 or 3 at one time? What are the outcomes for these various scenarios for the simulated payload plasma levels?
So in the Simcyp simulator, it can just be one at a time. So it is not possible that 3 molecules will be cleaved at the same time. So that's a restriction in the simulation. It might happen under physiological conditions. I guess to have such kind of scenario, we could still arrange the deconjugation rate and look what happens to the plasma levels. But I guess if you have a very quick release of all your different payloads, then and you have high permeability, then or it happens in plasma itself, then you have high concentration. Ideally would be you cleave it and you have it in your tissue without moving into plasma, I guess.
Awesome. Well, thank you so much, Himanshu and Felix. And those were some really excellent questions from our audience. So thank you to them for their great questions and their attention. Felix, can you go to the last slide, please?
Yes.
Thank you. So just want to share with everyone the next webinar in the series, which will be February 24th next week. We are partnering with NASDAQ, and our very own Dr. Julie Bullock will present what oncology drug developers should expect from the FDA's Project Optimus. Visit us at Certara.com to register for this and other upcoming webinars. And on behalf of Certara, I'd like to thank our presenters, and I'd like to thank you, our audience, for attending today's presentation. This concludes the webinar. Goodbye and have a great day.
Thank you.
Goodbye. Thank you.